Hello, everyone. My name is Zhong from Scientific Engagement Team. Today, I'm very happy to have this chance to share my knowledge about IO drug development. My presentation title today is Accelerate IO drug Development with a fast, cost-effective, and a powerful screening tool, MuScreen. In this presentation, I will discuss why syngeneic model plays the central role in immunotherapy development. What is Crohn's MuScreen platform, and how MuScreen can benefit our IO drug development. When develop a new drug, we needed to prove the efficacy and safety in animal before doing human tests. Regarding immunotherapy, we need to use animal model with a functional immune system and to assess the tumor interaction with it. There are two major kinds of model often used: human-based model and mouse-based model. Let's talk about human-based model first. This system combines human tumor, either CDX or PDX, and human immune cells. The immune system can be reconstructed by human PBMC or human hematopoietic stem cell, HSC. This platform can instinctively respond to human biologics like antibody, and it keeps the human feature of the tumor and immune system. However, it has some big drawbacks, such as not having a complete immune system, causing GvHD, depending on donor immune cell capability, and being expensive. On the other hand, mouse-based model has a natural, fully functional immune system. The tumor part can be syngeneic cell line or syngeneic homograft-derived models. Due to a comprehensive mouse tumor microenvironment, immune cells, and the stroma interface, this model can show how the tumor microenvironment affects the immune response and how the immune memory develops. This model can also help us understand the potential side effect of immunotherapy. Mouse-based models are easier and cheaper than to run the experiment than human-based models. Of course, the syngeneic platform has some limitations. For example, the mouse model have limited relevance with the human model, and having limited a model number we can choose for research. And the mouse model also has the variable tumor growth, but it is still valuable for drug early development. It will be interesting to discuss the translational meaning from mouse syngeneic model to human for IO research. Many studies have shown that mouse models are not very similar to human regarding some cancer types. For example, some mouse liver cancer model, like the HEPA-16 or H22, responds well to immunotherapy, but not in human case. And some mouse myeloma model, like B16F10, does not respond well to immunotherapy, but humans do. By this, we will ask why we are using mouse model to do immunotherapy development. As this paper said, when we looked at how the tumor and the immune system interact, we can see mouse and human are more alike than we think. And we did see some evidence by our data. When immunotherapy works well, in a mouse model like HEPA-16, it makes the immune system more active and less suppressive inside the tumor. It is also what happens in human. This means immune tumor microenvironment is more important than the cancer types for a preclinical research. So when we think about validate a new concept of immunotherapy, mouse syngeneic is the platform we should consider in the very beginning. It will help us to understand how immunotherapy works. Now let's have a break and look at the landscape of immunotherapy today. These data are from active clinical trials. Up to 2022, over 60% of active immune-related clinical trials were testing checkpoint inhibitor. PD-1 and PD-L1 are no doubt the most important one, occupying more than 70% in immunomodulator trials. But we cannot ignore there is a rapid rise of other types of immunotherapy, like cell therapy, T-cell engager, vaccine, and so on. We can see the PD-1 combo therapy has rose dramatically in the past 5 years. Interestingly, we were seeing a significant increase in PD-1 combination with either chemotherapy or VEGF-related therapy. But the new trial using CTLA-4 and PD-1 combo therapy decreased a lot. Now, let's talk about what the syngeneic platform can do around the current status. As we discussed a moment ago, the single agent and the combination therapy can be tested in this syngeneic platform, such as the novel checkpoint inhibitors, vaccine, oncolytic virus, microbiome modulators, cell therapy, immune modulating ADC, and the cytokine, and so on. For combination use, we can test the more than 2 different checkpoint modulators together, and also immunogenic cell death inducer, like the chemotherapy or radiotherapy plus immunocheckpoint inhibitor. Also, we can test the novel therapies plus ICI. Over the past few years, ProntBio assists our clients with various modalities, including all the types we mentioned here. If you are interested in learning more about our experience, we are happy to arrange another meeting with you to discuss that. And the biomarker discovery is another very important application field. I will introduce it in the latter part. As we discussed, IO is studying the interaction between tumor and the immune system. If you have a syngeneic collection cover diverse immunotherapy response and represent various immune tumor microenvironment interaction, it will be an ideal platform to test our novel IO drug. So we tried the classical immunocheckpoint inhibitor, including PD-1, PD-L1, and the CTLA-4 on our syngeneic cell line derived models. We realized we have 12 popular models indicated by these arrows have very different level of tumor growth inhibition after treating the 3 different checkpoint inhibitors. Next, we looked at the tumor-infiltrated immune cells of these 12 popular models. The immune profiling of each model is quite different, including the infiltration ability, effector cells percentage, and the suppressive cell ratio. Later, we set up the industry-first in vivo screening platform for in vivo oncology immuno research purpose, called MuScreen. It has above-mentioned 12 popular cell line-derived models and additional 6 popular homograft tumor models. It is an excellent option for the clients in early-stage drug efficacy screening or biomarker and mechanism discovery. We routinely run all screen models every quarter, and the clients share the vehicle groups and only pay for their own drug groups. QuantBio will provide the TGI results in the efficacy mode and analyze the tumor or tissue samples for immune cells, either by RNA-seq or flow or other method in the PD mode. And the clients can also ask for further analysis if they want to understand the mechanism better. So with our screen collection, you can save time and money and learn more about your drugs. Here is our screen collection of syngeneic cell line derived models. We now have the immune profiling data, including the TILs, spleen, tumor-infiltrated lymph node, and blood. And the baseline genomic, transcriptomic, and proteomics data are also available in our online database now. In addition, we measure the major cytokine in the serum of each model. And we check the typical TME markers by IHC. We also detected how the immune cells changed over time after PD-1 inhibitor inhibition. So we can share this data if you want to know it more, so you can feel free to contact us to have that. Here, I would like to introduce the syngeneic homograph models. This is another very useful platform for immunotherapy research. QuantBio calls it MuPrime. It is well known that syngeneic cell line derived models do not provide the clear information on on-call driver mutations, but our MuPrime model do. Let me tell you how we make MuPrime first. In the beginning, we use transgenic mice harboring specific on-call mutation gene or treat them with defined carcinogen. Then these mice got spontaneous tumors. Later, we collect the primary tumor tissue and then transplant it into naive stem strain mice, just like what we do in the PDX model. This model have clinical relevant driver mutation and much fewer total gene mutation, just like the human tumor feature. This makes syngeneic homograph model close to their human counterparts, and it is very useful for developing immunotherapy for different cancer types, especially with hotspot mutations. We here selected 6 popular homograph models from 6 different cancer types and provide the benchmarks for immunocheckpoint inhibitor treatment, immunoprofiling, genome-wide genetic features, and some representative protein staining results. In the next part, I will introduce 2 application cases with the screen. The first one is a proof concept research. We use screen platform to assess Tesaro in investigating their PARP1 inhibitor, niraparib. The PARP inhibitor activates STING signaling and induce the expression of type 1 interferon. This activation should enhance the tumor immune response. So we want to validate that. We tested the combination therapy of niraparib and PD-1 through our Millscreen models. We found synergistic and additive effect after the combination therapy in both BRCA deficient and BRCA proficient models. In this case, Millscreen platform demonstrated the possibility to enhance PD-1 therapy by PARP inhibition. For the second application case, I will show you how we use Millscreen for biomarker discovery. Millscreen provides baseline multi-omics data, including gene and protein expression, as well as the immunocheckpoint response results. We performed a comprehensive for biomarker discovery based on this data. And this data also can predict the clinical response and help us to find the right patient in clinical trials. First, we define the responder and non-responder for Millscreen models by their TGI results. Then we used a tool called MOFA to analyze our data and find the hidden factor that related how well the drug works. We found factor 1 is very important for all 3 immunocheckpoint inhibition factors. This difference is especially clear for CTLA-4 treatment. You can see here. It is consistent to the heat map score. Finally, we want to know what is the biological meaning about factor 1. So we check what gene and protein were involved in it. We found that factor 1 indicated higher activation level of IFN gamma alpha response, JAK status signaling, and complement activation. By combining Millscreen and bioinformatics, we can discover these potential biomarkers for immunotherapy. Before I end this presentation, I want to summarize the main points. Different immuno-oncology models fit specific application scenarios due to their unique features. Millscreen model offer a wide range of immuno-oncology interactions supporting mechanism of action studies. The Millscreen panel is highly capable of discovering IO biomarkers when used in conjugation with baseline data or downstream analysis. It is all of my presentation. Now I'm happy to take any of your questions.